Monohybrid and dihybrid crosses, first of all. So spec points to be very familiar with. And so monohybrid inheritance is basically studying the inheritance of a single characteristic that is controlled by one gene with two alleles. Two alleles. If it's three alleles, then we're talking about a different scenario, right? And then we've got things like codominance and things like that. That doesn't mean that there can't be more alleles out there. So if we think about like eye color, you've got alleles for brown, blue, green eye color, but each one person is going to have two alleles only, right? Because we've only got two sets of homologous chromosomes. So the monohybrid inheritance is when we decide that we are only interested in studying the inheritance of a single trait, single characteristic. We've got dominant alleles, and they are symbolized in a capital letter. Recessive alleles are lowercase. You know that already from your GCSE even. And the genotype is the combination of the two alleles that an organism has. The total combination of the alleles. The phenotype is what is expressed, what is going to be the final observable characteristic that is the result of the genotype, but also the environment. And the phenotype, if we have a recessive and dominant allele, if there is at least one copy of the dominant allele, the organism will display the dominant phenotype. So let's see how we do a simple monohybrid inheritance. So in this particular case, sorry, I forgot to say that the capital G is the dominant phenotype for a green pea plant, and lowercase g is the recessive allele for a yellow pea plant. So using this classical Mendelian example, let's see how we can calculate the offspring ratios. We use this thing called Punnett squares. You've done this since year 9 probably. And so first step is to determine the parental genotypes and the gamete genotypes to put into the Punnett square. So basically, one thing that you can bear in mind is that when we are, so for example, let's say that one parent here is GG. That is their one parent's genotype, right? They're heterozygous for this trait. When we are looking at this, when we're looking at a heterozygous parent, in our minds, what do we translate it? Oh, in their somatic cells, they've got one chromosome that corresponds to, that has this allele. They've got another chromosome that is this allele. These are homologous chromosomes. Remember, homologous chromosomes are separated from each other in meiosis. So the gametes that I'm going to write here basically represent the products of meiosis, right? Which is, and gametes are, of course, haploid, N, not 2N. This cell is 2N, diploid. Gametes are haploid, so they're only going to contain one homologous chromosome with whatever allele that carries. So half of this parent's gametes are going to contain this allele on the one homologous chromosome, and the rest of them are going to contain this allele on the other homologous chromosome. So let's say that, for example, this is the female parent, and the male parent is also heterozygous for this trait, and here are the other parents' gametes. So then we just literally go ahead and fill in the Punnett square, determining the possible genotypes first of the offspring. Sorry, just fix my chair. 1 second. The possible genotypes of the offspring, which would be homozygous, dominant, heterozygous, homozygous, recessive. And then we can calculate the phenotypes. Let me change color here. So the phenotypes would be 1, 2, 3, the dominant phenotype, so green, and 1, the recessive phenotype, so yellow. So we call this the ratio of offspring phenotypes. We often call the offspring the F1 generation, stands for filial. F1 generation, this is the parental generation or P generation. What if we have codominance? So this is the sort of simple, straightforward scenario where one allele is dominant over the other. What if we have codominance? Codominance is when two alleles can both be expressed at the same time without one of them being dominant, and the final phenotype is going to be a mixture, a mixed phenotype of both. So let's consider the following example. Where, and we are going to write the genes, the alleles, a little bit differently here. So we'll usually write like a letter with a superscript on the allele, just to kind of indicate that there's codominance happening. So let's, for example, consider this example of flower color. So we've got this allele codes for, so PW codes for white flowers, and PR codes for red. And there is codominance between those two alleles. So it's easy to imagine that actually the resulting phenotype from those is going to be a mix of white and red, which is pink, right? So let's go ahead and fill out this Punnett square. So we're going to have here PWPR, dominant for, homozygous for red. It's supposed to be a P here. Homozygous for white. Heterozygous. I don't know why I keep doing, sorry. PR, right? So let's now look at the phenotypic ratios. The phenotypic ratios are going to be 1 white, 1 red, 1 white, 1 red, and 2 pink, 1 pink, another pink. We sometimes write this ratio as the sort of 1 to 2 to 1 ratio, but even if you write it like that, it's fine. Another example of codominance is the human blood group, which is determined by 3 different alleles. One allele codes for antigen A, one allele codes for antigen B, and the other allele codes for antigen O, which in fact an absence of antigen. And so if we cross here, for example, an individual, so and B and A are dominant over allele O, but they are co-dominant to each other. So if we fill out this Punnett square, so this is an individual with blood group B, this is an individual with blood group A, right? Because A is dominant over O, B is dominant over O. And so here we have an individual that has these 2 alleles, right? So what are the phenotypes now? Let's interpret the phenotypes from this ratio of genotypes. This person is going to have blood group AB because A is co-dominant to B. This is gonna have blood group B because B is dominant over O. This is going to be blood group A, and this, of course, is going to be blood group O because I've got 2 copies of the recessive allele. If you're EdXLA, you don't need to know this stuff. Let's have a look at an example question. So in a monohybrid cross, what is the expected ratio of the various phenotypes when heterozygous parents are crossed? You don't actually even have to write out the full Punnett square to do this one, because it's just such a classic ratio. You're going to end up, memorizing it effectively. So our context are our context are the phenotypic ratios, right? In a monohybrid cross. My command ratio, my command word is that I only want a ratio. So the ratio is actually my command word. And my direction is that I need to determine both the phenotypes, the genotypes, and therefore the phenotypes. So what is the ratio of the various phenotypes? Oh, sorry, I wanted to change color. I thought I had. The phenotypes from a het cross, from a het cross. So, oops. So how do I solve that? If I actually go ahead and do a Punnett square like we did right in the beginning, the ratio are 3 to 1, and this will be the dominant phenotype, and this will be the recessive phenotype.